some ongoing work and some really novel insights on the short-nosed work.
Right, I actually thought you were going to be much meaner. So now I feel a little bit better. Yeah, so apologies to anybody who thought they were going to get a twofer for Amanda, but get me instead. Like Dwayne said, over the last couple of months, I have had the pleasure of working with Amanda, though, and not the pleasure of working with Dwayne, but we all do things we don't enjoy. But we have been kind of doing some extensions to the work that Amanda just presented. These new projects, they're going in a lot of fun, different directions, but like Dwayne said, the project I'm going to present today is one that we are using those acoustically tagged short-nosed sturgeon that Amanda just talked about to gain some insights into habitat use down in New York Harbor. So, as Amanda just kind of talked about, there's obviously a lot of reasons to celebrate the short-nosed population here in the Hudson, right? So, sturgeon, they are one of the most imperiled taxa in the world, so to have here in your backyard a population that is stable, increasing maybe at 70,000 fish, that's pretty damn good, right? But at the same time, we still don't know a lot about life history and ecology of short-nosed sturgeon. The last kind of species reports, especially specific to the Hudson, those came out a couple decades ago, and in that time we've obviously changed the anthropogenic stressors that we are putting populations under. And as this population has expanded, we should expect that its ecological niche may have also kind of evolved to use new habitats and to, behave a little bit differently than they may have been 30 years ago. So because of these uncertainties, though, we have a lot of issues about trying to figure out what are our best strategies for conserving and protecting short-nosed sturgeon. And really the question is, yes, this population's recovering, but how, right? What are the mechanisms ultimately responsible for this increase in population size? So as we try to get a feel for this question, I think, as Amanda kind of mentioned, one of the things we start looking at is like habitat, right? Like a fish needs really good habitat in order to survive. So obviously we know Esopus Meadows, that's a good overwintering site. They spawn up in Albany. These are known areas. But we are starting to kind of turn our attention to other areas of the Hudson that might also be critically important for short-nose. And so when we do that, we do start to think about New York Harbor. There are some kind of anecdotal reports of short-nose being down in the harbor, but we don't really know when or why those fish might be down there, and that's kind of problematic, right? Because the harbor, it's basically in the backdrop of one of the most urbanized landscapes in the world. And there's all these issues potentially with, ship strike mortalities and river construction or even offshore wind. So we really do need to get a good handle of if and why those fish are down there in order to kind of mitigate some of these threats. Like I said, as we started to near the end of that abundance project, we did feel like an opportunity had kind of fallen into our laps to use those acoustic tags to kind of start addressing this question and see if and when those fish are down there. So Amanda talked a little bit about our acoustic tagging process. We started with 100 fish, but we lost about 16 of those adults to just tag drop, probably maybe a little bit of mortality. But we were left with 84 adults, and importantly, we had a pretty good bias in our sex ratio of those tagged fish. About 3 to 1 male to female, and that's going to become important here in the next couple slides. In terms of our telemetry receivers, we had up to 7 receivers down in the harbor. It's actually technically Upper New York Bay, but I'm going to keep calling it the harbor because it just is a little bit easier to say for this talk. So those 7 receivers, they are still out there collecting data for us, but for this talk, we're just kind of truncating it to spring of 2022 to fall of 2024. And then in terms of our analyses, nothing crazy or complex, no Bayesian mumbo jumbo. We just really are mining that detection data to get at like average arrival and departure times, and then hopefully get an idea for like individual residency. Just again, when are these fish going down there and how long are they staying? So when we do that, what we see is that about 30% of those tagged fish, they do go down to New York Harbor. It's a little bit hard to see these colors, but this figure here, we have date on the x-axis, and on the y-axis is each of those 3 years of data. And the color indicates the proportion of harbor migrants that were detected in any given day of the year. And those red or warmer colors, those are just higher proportions. And so what you see pretty immediately is the highest number of fish that we're seeing in the harbor. They tend to go down in about mid-May, so we see a pretty strong indication of a spring migration for these short-nosed sturgeon. These spring migrants, they typically arrive in mid-May, they stay about a month, and then swim back up to the Hudson River sometime around mid-June, early July. And if you might remember, a couple minutes ago I told you that our tagged fish, they were kind of biased towards males. What's interesting is that our harbor migrants actually have nearly an equal sex ratio. It's not statistically significant, we don't have great power with these low sample sizes, but it is starting to get me thinking that the probability of harbor migrants is probably a little bit higher in females, but we'd probably need more tags to really tease that apart. So after you look at that spring migration, the next thing that kind of jumps out at me is that we see this extra little uptick in detections in about mid-September. And that's because we see a subset of fish actually making a fall migration back down to New York Harbor. So these were fish that often made that spring migration, went back up to the Hudson, turned right back around, and came down around mid-September. These fall migrants, once they were down there, they did have like a pretty variable duration of occupancy. Some fish stayed a couple days, some fish stayed a couple months, and then we actually had some fish that just totally overwintered down in New York Harbor. These fish, they would stay kind of in the harbor or adjacent habitats all the way through June of the next year, so it was like 9 months of continuous duration of occupancy down in the harbor, which is pretty long. So, we get to hear, you might be thinking, this is cool, right? There's some fish going down to the harbor, that's fun, but it's only 30%. But I do just kind of want to do a quick math equation here, where, if Amanda just said the population was 60 to 70,000 fish, 30% of that is like 20,000 fish that are probably down in New York Harbor, mostly in May. And so there's a lot of fish down there that are potentially running into all these threats that I mentioned earlier. So you start to think that, the harbor is obviously accumulating short-nosed sturgeon pretty quickly. So this project, it could have basically stopped here, right? Like we went into this, we just wanted to document arrival and departure, no big deal, mission accomplished, right? But when we started to look at the data, we realized the story is actually even more interesting than that. So the plot here on the right, it is a detection history for one individual fish, fish number 12, where we have date on the x-axis, and on the y-axis is just the river kilometer of that detection. And what I want you to notice is the fish is up here above RKM 200, so that's where they're spawning at. And then it basically immediately hightails it down to New York Harbor. Within a couple days, it's going from Albany all the way down to New York Harbor. So that's pretty cool, right? It's going really fast, but what we found interesting is that it's actually going to the harbor for 3 consecutive years. So that's curious, right? But fish do weird things, that's not all that crazy. But when we looked across all of our harbor migrants, what we saw is this really high repeatability in that migration strategy. So for this figure, I have again date on the x-axis, and on the y-axis is individual ID. And the way to read this, the colors are a little bit hard to tell apart, but the way to read this is that left to right, the color of the plot just indicates a fish's daily location through time. So just to display that a little bit better, this is fish number 12 again. It's the same fish I just showed you in that previous slide. You can just see, again, in 3 years of our study, it migrated to the harbor from about May and was there through about mid-July. So looking across all of our fish again, what we see is that if a fish went to New York Harbor in 2022, there's a pretty good chance it went there in 2023 and 2024. So what we start to think is this repeatability in behavior, it's not just like a random subset of fish that are going to New York Harbor, it's probably indicative of some sort of like cryptic life history diversity, where there's a subgroup of short-nosed sturgeon that really critically depend on occupying habitats in the harbor to complete their life cycle. The other thing I want to point out, though again the colors are a little bit hard to see, there's some fish I have here in blue. Those are fish that were not detected for at least a month in our array. We think that those fish probably migrated out of the harbor, potentially either to Newark Bay, Long Island Sound, maybe to lower New York Harbor. But we are installing some receivers this spring to try to get a better handle of kind of these possible migration corridors to try to narrow down whether we are losing some fish out to the ocean. But you can see they come back, so we're not losing them. But anyway, interesting migrations down there. So now that we know that those fish are down there, we start to wonder, what's the ecological significance of those harbor habitats? We think, we don't, these are all hypotheticals right now, but we do think that, lower estuaries, particularly those in urbanized settings, they can be really productive areas, so it's probably a good forage opportunity. There's also the possibility that there's increased water temperature in spring or in fall, which can just lead to more efficient bioenergetic processing. Again, all of this is kind of hypothetical right now because we don't have great data to really support some of these claims. But I will say those harbor migrants did tend to be larger on average than our river residents. So it does suggest that there might be some sort of fitness or fecundity advantage to migrating down to New York Harbor. The other question that we start wondering is, what is actually maintaining this life history diversity? Since we seem to have two groups of fish, harbor migrants and river residents, what's maintaining these two different phenotypes? And it would be really great if it's genetically based, because that would be pretty easy in terms of like an explanatory mechanism. I just don't think it is. Because all of these fish, they were tagged at the same time of year up in Albany, and so if there was truly two genetically divergent phenotypes, they'd have to be kind of partitioning their spawning resources at really, really fine temporal or spatial scales. So it's not totally out of the question, that's just not usually what drives total genetic divergence. At the same time, genetics is obviously a fickle beast, so it still could be genetics. It's just probably going to be something that's a little bit more nuanced, like a single gene that controls this, maybe some sort of genotype by phenotype interaction, or some sort of epigenetic mechanism. The other reason I think that there might be some sort of genetic control is that, if you remember earlier, I showed you how those fish were rapidly going to New York Harbor. If you think about it, that's a crazy physiological mechanism to go from full freshwater to full saltwater in the span of a couple days. So just physiologically, these fish seem to be a little bit more plastic in their osmoregulatory processes, suggesting again there might be some sort of genetic underpinning to this behavior. Big picture, I think what it really is just pointing to is that we need to do some future work, hopefully concurrently, to look at how fish are using those spawning resources. Is there some sort of fine-scale division there? And then hopefully also do some genetic and genomic studies to see if there are some biological mechanisms for explaining these behaviors. So kind of stepping back out as I kind of wrap up this talk, I think that big picture we are seeing right now, the harbor is an important and critical habitat to Shortnose sturgeon, and they are definitely using it as a piece of the mosaic to complete their life cycle, at least some proportion of them. And so that's really cool, right? We now know a lot more about Atlantic sturgeon life history and ecology, so that's great, but it immediately makes us start to wonder, is that habitat actually an ecological trap, right? Are fish either going down there because there's, some sort of ingrained behavior that they're doing, or potentially they're being motivated by a food resource? Either way, are they getting down there and then, potentially being exposed to all these anthropogenic stressors once they're there and having some sort of increased mortality? And so I just kind of wanted to think about, exactly how, it's a huge population of short-nosed sturgeon, so why would we care about threats in the harbor, right? So if you think about it in terms of just a pure numbers standpoint, we're looking at 30% of that population, about 20,000 fish, that are going to be directly in harm's way of any threats that are encountered in the harbor. In terms of where these threats are kind of spread across the population, it's obviously not going to be all of the fish that are affected by these threats. It's going to really be targeting those harbor migrants. And whether or not migration strategy is a phenotype or a genotypic control, what we would expect is if these threats are really targeting these harbor migrants, then we're going to have declines in either phenotypic or genotypic diversity. And so big picture, any threats that do encounter in the harbor, we have this risk of not only decreasing overall population size, but we also start to run the risk of decreasing resiliency through loss of things like functional diversity as well. So with that, I'm sure I have some time for questions, but if not, obviously feel free to reach out to one of us, but choose wisely when you decide which of the three of us to engage with. Thanks.